Hi, my name is Jacob Houghton, and I'm an assistant professor of radiology at Stony Brook Cancer Center and part of the research program for imaging, biomarker discovery, and engineering sciences. Today I'm going to be telling you a little bit about some of the research we're doing in nuclear medicine for personalized cancer care. I'm going to start today by telling you a little bit about radiopharmaceuticals. So radiopharmaceuticals are a way we can deliver radionuclides very specifically to cancer cells. We want to do this for a number of reasons, including imaging of disease for diagnosis, staging, or predicting or monitoring response to therapy. And in some cases, we're using them for targeted therapy of disease with or therapeutic radionuclides. And to kind of orient you, I wanted to first go over kind of the anatomy of what is a radiopharmaceutical. So in our case, my lab is working in cancer. Typically what we're doing is targeting cancer cells, and specifically we're targeting a protein, or also known as an antigen, on the surface of cancer cells that is very specific to those cells and is not present on healthy cells. The way we do this is with something called a targeting molecule. There are a number of different types of targeting molecules we could use. They could be peptides, they could be small molecules, or in the case of my laboratory, we're typically using what we call antibodies. We then use what we call a chemical linker to attach something that allows us to incorporate a radioactive isotope. There are a number of different radioactive isotopes and different applications for those isotopes. And my lab is particularly interested in isotopes for positron emission tomography, or PET imaging. And the reason that we're interested in PET imaging is that scans can provide very important information about the diseases, particularly cancer, and it does so non-invasively. So this is different from other types of procedures that you might think of as like a biopsy, where you're actually going invasively in and looking at the cancer. We can get the same type of information non-invasively using a PET scan. And these PET scans can help physicians to make decisions about patients' care. And those decisions might involve what type of therapy would be most effective for their disease, the severity and location of the disease. They might use it to follow response to different types of chemo or immunotherapies, or to look for recurrences in patients who have already undergone treatment. And the way this works is that we're targeting cancer cells that have very different biochemistry than our normal healthy cells. And we can actually design these radiopharmaceuticals so that we can target those differences very specifically. And the radiopharmaceutical then that we have designed, once injected into the patients, will accumulate in the cancer cells and not the healthy cells. And then the nuclear medicine and radiologists are able to actually read and interpret the PET scans and tell very important information to the patient's physicians regarding their disease. So one of the primary uses for PET imaging is to support cancer immunotherapy. So I want to tell you a little bit about what cancer immunotherapy is. It's a rapidly growing treatment option. There's been over 100 FDA-approved immunotherapies in the past decade, and it differs somewhat from traditional chemotherapy in that it is, again, very specific. So traditional chemotherapy typically targets some sort of DNA processes that goes after rapidly dividing cells, as we know cancer cells typically are. But this indiscriminately kills these cells, whether they be part of healthy tissue, think maybe hair follicles, which can lead to hair loss during chemotherapy, and as well as the tumor cells. Immunotherapy, on the other hand, targets biochemical changes in tumor cells, and only those biochemical changes in the tumor cells, sparing the healthy cells and allowing us to target the disease very specifically. And this is typically occurs through the use of monoclonal antibodies. So I won't go too much into the details of what monoclonal antibodies are, but I do want to say that these are antibodies just like your immune system uses. We can actually engineer them, and what's really nice about these is that they're developed to target the biochemical changes or the antigens in the cancer cells very specifically. And this high level of specificity allows us to target those changes and only those changes at the disease sites. And what's even nicer about this is that we can actually engineer these to then, once they get to the target cancer cells, to engage the patient's immune system in a way that allows our immune system to naturally attack the cancer cells, sparing the healthy cells and using our natural immunity to actually fight off the cancer. So immunotherapy, as I've mentioned, has been around for decades now, but it doesn't work for every cancer. And that's because cancers are different. Not everybody's cancers are the same. And that means that immunotherapies aren't always going to work for everybody because there are thousands and thousands of possible mutations, and there's even larger number of combination of mutations that can occur in cancer. This is what we call cancer heterogeneity. So basically the main point here is that no tumors are alike, And this actually makes it somewhat difficult to effectively employ immunotherapy as kind of a blanket type of treatment for many cancer patients, which is where my research comes in. So I'm interested in what we call personalized medicine, which is defined by the American Medical Association as a way of basically separating patients into different groups with their medical decisions, the practices of the physicians, their interventions, and the products that are being tailored to the individual patients being based on their actual predicted response or risk of disease. And so the way that my lab is contributing to personalized medicine at Stony Brook is by developing these PET imaging agents that I've told you a little bit about. And so kind of to explain how this works in practice, is you can imagine that we have a large patient population of, say, patients with lung cancer. Everybody's cancer is going to be a little bit different, but there's going to be a lot of people that have very common mutations in lung cancer. And luckily, there's a lot of companies out there that are developing these immunotherapies that target these very common mutations. What my lab can do is use PET imaging based on those antibodies that have been developed for immunotherapy. And we can actually take the general patient population and screen them. And what we do is we can stratify them into people that have a lot of this particular biomarker and those that don't. And those that don't, unfortunately, are most likely not going to respond to that type of immunotherapy. However, those that do can then receive what we would call a personalized therapy. So we know these patients are expressing the biomarker or antigen of interest on their cancer cells. So what we can do is, after we've separated them into potential responders and non-responders via PET, they can go ahead and receive that therapy. And then we can follow up with them later on, again, using the same type of PET imaging. And we can actually look at those who are responding and those who are not responding. And where this becomes important is, especially in clinical trials, is predicting responders and then moving patients off of these drugs that aren't going to help them. It's great to help those, and show that we can help those that are going to respond, but it's really important to actually separate those that are not likely to respond so that they can get on a treatment that will actually work for them. And so how my lab is assisting this process is developing monoclonal antibodies that are labeled with PET isotopes for personalized immunotherapy. I won't go too much into the technical details, but basically what we're doing is we're taking a radioisotope called zirconium-89, and we're doing some chemistry on these antibodies that are generally approved immunotherapies, and we're attaching this to them so that we can use them for PET. This isotope, 89 zirconium, emits positrons that can be used then for PET imaging. And what happens when these things are going into humans is patients are administered a small and very safe dose of radioactive antibody. A lot of people get a little bit scared about the radioactive part of this, but it's typically no less or no more radiation dose than a typical flight across the country. And when we do this, then we can take these particular therapies and determine if a patient may or may not respond to them. And this could help us make decisions about, which patients should enroll in a particular trial. And then once we have these, the physicians can also use these for looking for things like, are there small metastases that we can't see? Most patients will receive either an MRI or a CT scan, but those are best for identifying your primary tumors that are somewhat large and you know kind of where to look for them. In a lot of cases, there can be small metastases that those types of scans might not pick up that we can actually see with our PET scans, which I'll show you a little bit on the next slide. And then again, we can use these to follow these patients. Is the treatment working? Are they responding? And if they have responded, is the cancer recurring later on? So there's a lot of different applications. And so my laboratory does the kind of preclinical research. We call what our lab does bench-to-bedside translational research. So we take these immunotherapies, typically a monoclonal antibody, and do all of this work that we've told you about, labeling them with zirconium, and then we typically test these in mouse models of cancer. And so I've kind of shown a very brief overview of kind of what that work looks like for us. In this case, you can see a mouse on this slide that has pancreatic cancer, which is in the red box. Unfortunately for this mouse, it has also developed liver metastases and as well as metastases in the lymph nodes, which you can see using our 89 zirconium monoclonal antibody. We're able to trace throughout the body. What we're seeing is cancer and only cancer and none of the healthy tissues, which allows us to very accurately, again, stage this type of disease. So this is very similar to what we would be doing in humans. But what we do in my lab is to develop this, do the chemistry, and then actually do all of this work in animal models to show and to prove to the FDA that this work is actually safe and could be done in humans. And the last part of what my lab does that I'll tell you about today is actually taking these same monoclonal antibodies and using them for a different type of therapy that we call targeted radionuclide therapy. So the premise of this is the same. We're taking an antibody that will go only specifically to the cancer cells in the body, but instead of attaching a radionuclide to it that can be used for PET imaging, what we're doing is attaching what we call a therapeutic radionuclide. So these are radionuclides that emit what we call alpha and beta particles. And while those are not able to be used for imaging, what they do is they cause damage to the cells to which these antibodies then bind. And so basically by replacing our PET radionuclide with one of these therapeutic radionuclides, we're able to deliver a therapeutic dose of radiation. And this is a somewhat new field, but one that is growing rapidly and showing really incredible results. And we're really excited to continue on with this research in the future. And I'd like to thank you for your attention. It's my pleasure to tell you a little bit about the research we're doing in my lab and at Stony Brook. If you have any questions, please submit them to coe@stonybrookmedicine.edu, and I'd be happy to answer them.